Hi everyone, welcome to this revision video on the structure of prokaryotic cells and viruses. This is based on the AQA A-level biology specification, specifically 3.2.1.2. Let's get straight into this video. So let's look over some key definitions that you need to know. Viruses are acellular, non-living particles. Prokaryotic cells are a cell without a nucleus and no other membrane-bound organelles. A mesosome is membrane infold, and plasmids are small circular loops of DNA, and acellular means that it is not consisting of cells. Moving on to some key terms. When we talk about the substance murin, we are also referring to peptidoglycan, and these are glycoproteins. So when we look at the organelles that you can find within prokaryotic cells, here's a quick overview. Prokaryotic cells are much smaller than eukaryotic cells. They also differ from eukaryotic cells in having a cytoplasm that lacks membrane-bound organelles, smaller ribosomes, no nucleus. Instead, they have a singular circular DNA molecule that is free within the cytoplasm and is not associated with proteins. A cell wall that contains murin, a glycoprotein, and in addition, many prokaryotic cells have one or more plasmids, a capsule surrounding the cell, and one or more flagella. So when we're comparing prokaryotes and eukaryotes, some similarities include they both contain cell surface membrane, ribosomes, cytoplasm, and DNA. But in prokaryotic cells, they are much smaller, whereas eukaryotes are larger. Prokaryotes lack a nucleus, whereas eukaryotes don't. Prokaryotes lack membrane-bound organelles, whereas eukaryotes have membrane-bound organelles. Prokaryotes contain smaller 70S ribosomes, whereas eukaryotes contain larger 80S ribosomes. DNA within prokaryotes is described as circular, naked, and free within the cytoplasm, whereas in eukaryotes it is described as linear, bound to histones, which are proteins, and within a nucleus. Prokaryotes contain a cell wall containing murin, whereas eukaryotes do not contain a cell wall containing murin, whereas plant cells can have their own one equally out of cells. And then some other organelles that you can only find in prokaryotes include capsule, flagellum, mesosomes, and plasmids. Moving on, let's look at a comparison of prokaryotes and viruses. So some similarities between the two is that they both do not contain nuclei, but some differences include that prokaryotes include a cell surface membrane, whereas viruses do not. Prokaryotes have a cytoplasm, once again viruses do not. And prokaryotes surprisingly do not have a capsid, whereas viruses do. Moving on, when we look at a comparison between the DNA within prokaryotes and eukaryotes, DNA within prokaryotes is described as circular, free in the cytoplasm, naked, whereas in eukaryotes, DNA is described as linear within a nucleus and bound to histones, which are a type of protein. And when we look at an overview of replication within these different types of, how would I describe them? These structures within eukaryotes, they follow the cell cycle, and within prokaryotes, they follow binary fission, and viruses, they follow viral replication. And today we'll be looking at the bottom two. So here is a quick diagram of a bacterial cell. Screenshot it if you need to. I think it's just like a quick way to remember it, and I think it's very succinct and actually quite good looking. No shade, my diagrams have been looking a bit dodgy as of late, so I'm really proud of this one. Moving on, let's look at a few structure and functions of some of these organelles. Plasmids, the function of these is used in genetic engineering, which is the final topic within this specification, which sounds like a ways away, but we will get to it in due time. But yeah, they are a key part of genetic engineering. Moving on to a cell wall, the structure within prokaryotic cells, which include bacteria, is that they are made of murin. And then the function of cell walls is protection, is able to maintain strength and shape and prevent lysis. We look at ribosomes, the structure of ribosomes within prokaryotes is that they are smaller, 70S, and they're made of ribosomal RNA and proteins, and their function is for translation/protein synthesis. And when we look at flagellum, the function of that is locomotion. When we look at circular DNA, these are described as having a structure singular, free in cytoplasm, and not associated with proteins, and their function includes the production of ribosomal RNA/ribosome, stores genetic information for protein synthesis, DNA replication/interphase, and the production of mRNA/tRNA/transcription. And when we look at the slime capsule, this structure is that it is a mucus layer containing water, and the function of this capsule is that it protects other cells/viruses from hiding antigens on the cell surface and allows them to stick with others, and it prevents desiccation. When we look at viruses are acellular and non-living. The structure of virus particles do include genetic material, capsid, and attachment protein. Genetic material which can include DNA or RNA. The function of that is that it contains the genetic code to perform new viruses. And when we look at capsid, which is a type of protein, its role is to protect the genetic material from the external environment. And when we look at attachment proteins, which no shock are proteins, helps viruses identify and bind to host cells to infect them. This includes HIV, which we'll be looking at later on within this specification. And we look at binary fission, which is the form of replication within bacterial cells, as we mentioned before, is a replication of circular DNA, replication of plasmids, and the division of cytoplasm to produce two daughter cells. Moving on to viral replication. Being non-living, viruses do not undergo cell division, instead undergo the aforementioned process where attachment proteins attach to receptors. The virus injects nucleic acid into the host cell replicates the viral nucleic acid, and the host cell produces viral proteins/capsids/enzymes. The virus particles are assembled and released from the host cell. And that is the end of this revision video. I hope this was helpful. Down below you can find the link to my revision booklets, which I think are incredible. I spent so much time on them. They are my heart and soul, honestly. I tried to make them as affordable as possible, so if you are able to support, please go and purchase them down below. But there are free resources on our TikTok page at all9GCSE guide. If you have any comments on it or any tips that you want to share with any other students, make sure you put them down below. And without further ado, happy revision, guys.